Our next speaker is Sheriff from the European Bioinformatics Institute. He will talk to us about biomodels and standards and reproducibility, a topic that really flows well after Maria's presentation. Thank you.
All right. Thanks for that introduction, Tom. It's a pleasure to be here and to talk to you about biomodels. It's been 20 years since biomodels started. It's been there in the community for a very long time. Of course, I had been with biomodels for half the journey, but I think it's a great resource, and I'm happy to share where we are right now. So biomodels, for those who do not know, is a repository of mathematical models of biological systems. I mean, modelers start building models, and they wanted to find a way to exchange models with each other, and they needed a platform exclusively for that, and biomodels came to existence to serve that purpose about 20 years ago. So it was built to allow a modeler to share their models in SBML initially, but then it evolved slowly to allow models in various formats. So you can have an ODE model, a logic model, a petri-net model, or constraint-based model. You can share all that with the community in any format. It can be in SBML, which is of course preferred, but it can be in MATLAB, or it can be a Python code or C++ code. It can all be shared through BioModels repository. And you can give access to a broader community to use your model. The way it works is, if you're a modeler, you build a model, you submit that to BioModels repository. It remains private, hidden, but then you include the accession number, unique accession number for the model in your manuscript. You send it to the review. When the manuscript is accepted and the paper is out, the model is also out in BioModels. Meanwhile, if the reviewers need access to the model, we can give specific access to the reviewers so that they can check the models directly from BioModels repository. So BioModels is a repository to share your models, but it's more than that, because we do extra value added service, which is the curation. What we do in curations, we have dedicated team members who basically look into the model, look into the paper, manuscript, and try to compare the results and see if all the equations are correctly encoded, there are no mistakes in the parameter values, initial conditions, and once they are happy, they encode these in SBML. SBML, for those who do not know, is systems biology markup language, which is a machine readable format to encode and share mathematical models. The reason we use SBML is it's highly interoperable. It's again a very long running community standard. It's almost as old as BioModels. So if you have your model in SBML, you can exchange easily with the community. And let's say I have a model I built in let's say in MATLAB, I can export that into SBML and then can share with someone else, and they can import it in Python, or they can import in R. And there are many tools like Copasi which can import SBML, and you can run simulations. So once you have your model in SBML, you can share this across multiple resources, and you'll be able to run these simulations. If you go back and take a SBML model from 15 years ago, 20 years ago, you'll be still able to run these models and get simulation results, right? Which is not possible for any other code. So if you go get a Python code from 15 years ago, 20 years ago, it'll be really hard to get it running. But with SBML, it kind of holds the essence of the model. And the SBML community always had maintained backward compatibility, that you can pick up any old SBML model that's curated and get the same results even now, which is really cool. And so once we have the model in SBML format, and we encode it in SBML, and then we run simulation in our preferred tool, for example, Copasi, and we try to reproduce the result. And this is very important for us. So for us, a model to be curated, called curated, we want it to be reproducible, and we should reproduce at least one figure from the original manuscript. If it is not reproducible, then we do not call the model curated, and we put them in the non-curated branch. Once we get the model reproduced, we add annotation and make sure that they are discoverable and findable through BioModels repository. I mean, we not just curate the models author summit, we also go and curate models from the public domain. In the past, we have collected and curated models in diabetes, neurodegeneration disease conditions, and also we have collected and curated a good number of COVID-19 transmission models. BioModels, of course, is very popular in the community for model discovery. For those who are modelers, who are looking for data, they look at PubMed, Google Scholar, and after that, BioModels is a popular avenue where they look for model-related data. And for submission is, of course, also well preferred after GitHub personal website and supplementary material. And BioModels content have been growing over years. It has grown further, and we cover various organisms as a big collection of models from humans, and of course there are also models from yeast and mouse, and also for many microorganisms. And these models broadly cover various aspects of biological processes and various disease conditions. This is like a quick snapshot, and of course this is growing and dynamically increasing depending on the submissions we get from the community members. Overall, BioModels right now has about 2,750 public models, and among them, 1,100 are about curated models. Most of them are kinetic models or ordinary differential equation models, and of course, a large fraction of models are still non-curated. This doesn't mean that they are not reproducible. This includes models that are not reproducible, also models that we haven't yet had time to curate. So we hope to curate them slowly, but there are also approaches where we still don't have standards to curate. So we're trying to build standards and make sure we curate and make these models reproducible and share them publicly with the community. And we also have a big collection of auto-generated models. This Path to Models has about 140,000 models. They are kind of bundled into 812 archives. And there's also a big collection of this patient-specific genome scale metabolic models. And we also have bundled them into 21 archives. And they're all available through BioModels repository. You can go to BioModels, search for any keyword. You can find the model of your interest and pick the model and explore it further and see if that's useful. And there are facets on the left-hand side, which allows user to kind of select various options and then narrow down the filters to find the model of their interest. Let me show you another example. So let's say if you search for COVID, you'll find all the COVID-19 models, and you can, let's say, pick one model, let's say a model of COVID-19 spread in Wuhan, and then get more details about this model. So if you go and click on this, you will be taken to a page where you can see a quick overview of this model, a quick description, a short description, a link to the original manuscript where this model is published. And also a bit of metadata on the right-hand side tells you a bit more about the model and what are the organisms involved, what approach is being used, and the identifier of the model, and so on. In the bottom of the page, you will see a bit of links to the external resources. And if you click on them, it will take you to other resources. For example, the SBGN will take you to the Newt editor, where you can visualize the structure of the model. And if you click on the Mendel-Macau platform, it will take you to this nice platform below a collaborator in EBI, which allows you to do live simulation of the model. So you have avenues to go to BioModels to find model, and then go to other resources to play and explore this model further. I mean, there are a few tabs I need to kind of explain you on how BioModels presents a model. After the overview page, you have this files tab, which kind of gives all the files related to this model. You can go and download any of these files, but the main file here is the SBML file that we curated, and we made sure that it is working and reproducing. So if you download the SBML file here, and import into any of these tools which support SBML, for example, Copasi, and run simulation, you should be able to reproduce the results in the original manuscript. And we also share the Copasi file, and sometimes we also have MATLAB file and other files, so you can quickly download them and reuse them. And if you go to the history tab, you'll have history of all the versions of the model, public versions of the model. You can go back and get the original version or the version which has been updated over time. And so you can track, it's all version controlled. And if you go to the components tab, it will quickly give you all the components that are relevant here. And you can also click on the links and go to the repositories where you can get more information about these annotations. And the last tab here is the curation tab, which is very, very important tab where we provide information about the curation. So here we actually show the figure that we reproduced independently, right? And for example, if you can see here, figure 4C has been reproduced. So we have reproduced other figures too, but we have showed here figure 4C. Sometimes we have to change certain parameters in order to get the figure working. We also mention this information here. And this is important process of our curation. Reproduce the figure and provide the evidence here that we could reproduce at least one figure from the manuscript. And this is very important for the user to get confidence and start reusing these models or component of these models. And I would like to also draw your attention to another nice external tool that's linked to BioModels, and that is called Talk to BioModels. And for those who do not know, it is like a chatbot, multi-agent AI-based chatbot, where you can talk to it and then ask it to do various tasks, and it will perform these tasks for you without having to go to BioModel or run these various simulation tools. So this is quite nice. If you're not an expert in modeler, you can say you can search for all the models in certain disease condition. You can download one of these models and ask it to run simulation for 50 days, whatever interval, and then run it to simulate various conditions. You can change the values. All you can do with the command line interaction and then analyze the model, which is quite nice. I mean, this is all possible because we do the curation in standard format like SBML, which allows you to integrate the model, play with. I mean, we also have APIs, right? Everything allows this user community to take more components of the biomodels, interact with the components of the biomodel, run simulations, and so on. So, thanks to standard again to let us do something nice like this. Also, because we have these models in SBML format, we can extract all the internal information, like what are the parameters, initial conditions, rates, and the metadata, and make them easily findable. So you can get these information through either a web interface or an REST API and use them for your own research. For example, if I'm interested in a protein, I can search for that protein in the BioModels parameters web interface. Is going to give me all the reaction this particular protein is modeled and the rate equations, and you can get more and more further information about this protein if you go and click on the external links on the right hand side. But it will take you to, for example, UniProt to give more information about the protein. You can go to Reactome to get more pathways information about this protein of interest, or you can go to SubURK if there are experimentally derived kinetic parameter values. So BioModels parameter is a good way to access parameters from BioModels repository. You can use it to scan or get the range of parameter values that are used in the literature in the models and BioModels, and you can use that as initial boundary condition to scan parameters in your model. You can use initial concentration to do the same, or you can take different components of the model, put them together, and run simulations as well to see how they help to enhance or build your model further. Overall, BioModels repository kind of provides fair and reproducible models. The models are findable, accessible, interoperable, and reusable, findable through various avenues. You can search the models through EBI search. You can find them through OmicsDI search engine. They're also pushed to Google datasets. So you can also search them in Google datasets. And they are all with CC0 license, highly permissible. People from industry can use it without having to worry about any restrictions. Everything's open source. Even the code base of BioModel itself is open source, which means anyone can take even the whole web interface and then create their own BioModels if they like internally. And of course, we focus on SBML, which makes things more interoperable. You can exchange models across various tools and run simulations, and it's reusable. Everything is curated, so you can reuse with confidence. And these are the two main reference publication where you may get further details about BioModels. Now I want to switch gears a little bit and kind of share with you how we kind of support models, but also improve the reproducibility of these models, right? So we know that there is this reproducibility crisis in the field, and not just one, but many fields of science. So that was the survey a while ago where they kind of did survey across experimental scientists to figure out if they could reproduce results. And 70% of them said they cannot reproduce, others is and about 50% said they cannot reproduce their own result. Right. And we know that from this, there is certainly a crisis in reproducibility in experimental data, I mean, from physics, chemistry, life science, and so on. But we wanted to ask do we have similar reproducibility crisis in computational modeling, resistance biology modeling? And we wanted to focus on improving reproducibility across various modeling fields. So let me go into a bit of details here and show what we have done to improve reproducibility of different modeling approaches. Let's start with ordinary differential equation models because that's the primary focus of BioModels for several years, and we wanted to see how reproducible ordinary differential equation models are. So before we go into reproducibility, I need to kind of give you a bit of background. There are two concepts. One is replicability, another is reproducibility. Replicability or repeatability is ability to rerun the same code in the same software and get the same results. Whereas reproducibility is the ability to recreate the code and run it a different tool and get the same results. So it's a bit harder and it's a bit more challenging and it's a bit broader than just running the same code. And our approach was to focus on reproducibility, not replicability. So we did this study where we kind of tried to curate about 455 models. They are coming from peer-reviewed articles from 152 different journals, published from 1980 to 2020. And of course, these models are not randomly picked. These are part of the regular curation we do in BioModels. And we try to prioritize in our curation based on various factors, collaboration, funding, and so on. However, this collection was a significant proportion of models and biomodels at that point of time. Right. So how did we do this? So we did the reproducibility assessment of each and every model. There's 455 models were individually assessed. And what we do is we make sure that we have everything in the paper. We encode the model in SBML, get the parameters from the paper or from the authors, make sure the model is properly encoded, and we run the simulation in copasi. If a figure is reproduced, then we know that the model is reproducible. It's great. If not, we'll try to do any minor fixes in the model and make it work. So we don't mind if there's a minor deviation in the simulation figure. At least one figure should be reproduced. Then we are happy. If the figure is too deviant and it kind of completely deviates from the scientific conclusion of the study, then we call it non-reproducible. However, if it is slightly different from the original figure, then we don't mind too much. So if a model is not reproducible, there are two approaches. One is empirically trying to fix the model, try to play with the parameter, with the equation, because you understand the description, you kind of know that that's a typo. There can be plus instead of minus in the model equations, or there could be a slight typo in the parameter value. Instead of 0.01, it could be 0.001. You can fix those errors empirically and try to see if it helps and reproduce the figure. If not, then you try to contact the authors and get information. This study was published a while ago, and many of you know about this. And the outcome was about half of the models cannot be reproduced based on the information provided in the manuscript. Right. Although these are mathematical models where we had mathematical equation described in the manuscript. So we didn't take those which had just the codes. We took those manuscripts specifically which had differential equations, ODEs, explicitly written in the manuscript. Right. And even in those cases, we cannot reproduce all of them. Only 50% could be reproduced without any further information directly. And of course, we try to fiddle with the model with empirical fixing. About 9% of the model worked, and then about 3% kind of worked with the help from author. About 37% of the model cannot be reproduced based on any information provided in the manuscript. So there's a large fraction of the model that remains non-reproducible. So when we try to contact the authors, this is some of the problem we face. A majority of them do not respond. Only a small fraction of them respond. Even when they respond, half of them do not provide useful information. They can say that we have left the group or moved on, and various excuses. So we cannot even reproduce even when they respond. We looked at the age and see if there is difference in reproducibility. The proportion of reproducible, non-reproducible model is more or less similar across all the ages, all the timelines we analyzed. And of course, we get more response from the authors from the recently published papers. And we looked at the journals, and there's no clear pattern between reproducible, non-reproducible models across the journals. And for some journals, we had very few manuscripts that we cannot label a journal good or bad based on this. However, we had a good collection of models where we can dive a bit deep and see what are the probable causes of non-reproducibility in these models. I mean, often you can see missing parameter values, missing initial conditions, or there is mistake in the structure, or a combination of these. However, for large proportion of the model, we do not know what the reason is. Everything is clearly written in the paper. We have clearly encoded the same. Still, the models are not reproducible. So these models, which we managed to reproduce as a part of the study, are now in the BioModels as curated. In the curated branch, those we cannot reproduce are in the non-curated branch. Of course, we haven't yet labeled them, named, and we don't want to name and shame. Of course, we want to label somehow that this model we attempted to reproduce, and it didn't work very well. Okay, so we have this problem. What is the solution? We came up with a simple solution. We came up with a simple reproducibility scorecard, which makes sure that if you have done everything correctly, sharing the model equation, sharing the parameter values, initial conditions, simulation conditions, all those things, so that it makes it easier for you to check these before you submit your paper and the model. And we've shown that this could be really helpful. We said if you get one yes, you get a score of 1, and if you get all 8 S's, then you get a score of 8. So as the score increases, we did some retrospective analysis on these models and showed that as the score increases, the models become more and more reproducible. Higher the score, better the models are reproducible. So we kind of suggest that at least a model should aim to have a score of 4, which kind of increase the odds of getting the model reproducible. This work is not new; it's been published quite some time ago, and I'm still always discussing this with people, and there are a few who do not know. So I would still want to remind people that this is a real problem which we need to address. Okay, now that I discussed about ODE models, now let's move on to stochastic models, right? And stochastic could be differential equation models. And we wanted to also improve the reproducibility of these stochastic models. And the problem here is that we do not have any approach. And then TJ came, and then we kind of had this brainstorming and discussion, and we came up with this idea of how could we compare two distributions, the stochastic distribution, and see if they are reproducible, which means there are two hairballs which is coming from these different trajectories, and can we combine them, so compare them and see who's reproducible, if they are reproducible. The problem here is that with the stochastic model is every simulation is a new result, so it is going to be stochastic inherently. For the stochastic simulation, we need to find a smart way to compare them. I mean, of course, people suggest that we can do summary statistics, but it's not accurate because you can see on the top right, you can have different distributions, and they all can have same summary statistics of mean and standard deviation, and they are completely different distributions. So you need to have a more clever way and to compare these two stochastic simulations and see if they are indeed reproducible. And also, I mean, it's not just reproducibility, you need something more than that to find if you're sampling enough. And we came up with this new approach called EFFECT, which is empirical characteristic function equality convergence test. It compares the ECF, empirical characteristic function, between two distributions and checks if these are indeed the same. Right. I don't want to go too deep. I might show a few slides afterwards into the math, but you can read the paper to get more info. And this helps us to understand, is the sample size of the simulation sufficient? And it kind of addresses this problem. How do I package this and share so others can test the reproducibility and let other gain confidence in the results? So this approach worked really well. We have a workflow where we recommend modeler to do first internal reproducibility check, where they kind of split the samples into two subsamples and see if the effect tells if the subsamples are reproducible. Then they share this publicly, and and then also this allows external curator or researcher to do the same. They generate another subsample, and then they can compare the ECFs from their subsample and the one from the modeler to see if the models are reproducible. Without going to details, this approach worked really well. Again, not going into details, and we could show that it can be very useful. It is method agnostic. It can be applied to ODE models where we do parameter sampling, stochastic parameter sampling, or it can be applied to GLFC based simulation algorithm of a stochastic simulation. It can be applied to agent-based model or Boolean stochastic model. And this approach worked really well and identifying when things are reproducible and things are not reproducible. And we try to see if to see if we can kind of analyze how reproducible stochastic models are. And we try to pick up analyze about 6 papers. Basically, TJ did this analysis and found that from the 6 paper, he compared 16 figures of stochastic simulation. And in 16 of the simulation, 14 of them are really questionable. Only 2, after lots of forgiveness, you can accept that they're reproducible, but a majority of these models are indeed not reproducible at all, which kind of tells you there is a bigger problem here in stochastic simulation reproducibility. It needs to be addressed. So EFFECT helps us by providing this new method that assesses reproducibility of stochastic simulation. we have a standardized way to export reference data set, and it also helps you to identify the sample size that's needed and also report a quantitative metric called effect error to quantitatively assess if things are reproducible. Okay, let's move on quickly because I'm running out of time. And there are other approach, right, in modeling, which is very common these days, is this includes a constraint-based model, which is very, very popular because you can easily generate lots of genome scale metabolic models. Again, here the generic problem was the solution space is much broader. You can have multiple solutions, and often people do not share all these solutions, which makes it really hard to test if a model is reproducible. So if you run this stochastic, not stochastic, the flex balance analysis here, you will have a very broad solution space, and this makes it really challenging. So we had to come up with this new approach called FROG, which lets you test whether a genome scale metabolic model is reproducible. What the FROG does is basically performs 4 different analyses and generates reference data set. This includes flux variability analysis and a single reaction deletion analysis and the biomass objective function and then also single gene deletion. So you perform these 4 different analyses, which provides you reference data set which is numerically reproducible. So what we recommend is here a modeler generates this frog report, which consists of this frog analysis outcome, and share it with the model. With this, anyone else can then pick the model and make sure that they could rerun these reference analysis and get the same reference data set and know that the model is indeed reproducible. Once we have that, then we can reuse this model or component of the model or, with confidence. And we have many tools supporting frog now. You can run frog very quickly through all these tools publicly available these days. Now we use these frog tools and see if the models are reproducible. Again, the constraint-based models are reproducible, genome metabolic models are reproducible. We had like a similar outcome. About 40% of the model are reproducible without any problem, and for the rest, we had some issues. About 28.6% roughly. Here we had to have some minimal intervention, and in some cases, about here 12-13%, authors completely submitted a wrong version of the model, right? So then we could ask them to submit the right model. Yeah, of course, there are some SBML related issues, software related issues, and thanks to Frog here, because it could identify the problem and tell where the problem is, and we could go back to the authors when needed and get the model fixed, or we can troubleshoot ourselves and get it fixed and get all of them reproduced finally. And here we could do this because all these models are submitted by authors of this Frog manuscript, because we are all together. Everyone helped, and we could get this thing working very soon. Right, so now coming to the last bit, the machine learning models, which are getting very popular. Some BioModels wanted to help in this space as well, at least provide some guidelines and help if we can make these machine learning models more reproducible. And they are very scattered, as you know. It's hard to find them and reuse them, and lots of missing information, data, and so on. So what can we do? So we apply the same pipeline we have for other models. You can submit a model, ML model to BioModels, try to reproduce it, add metadata, make it findable, discoverable, disseminate them again to BioModels. Makes sense. But then our criteria for curation includes reproducibility assessment, right, which is very strict. And when we try to assess the reproducibility, then we had a problem. So about 75% of the model in the initial study, we cannot even attempt to reproduce. Only 25% we could attempt to reproduce. What I mean here is that there is lots of missing component. That 75% model, you cannot even try and get them working, or there is major component missing in those. So even when you have everything, that's only 20%, 25%, only 10% is reproducible. Another 15% is not reproducible. So that is a bigger challenge here with machine learning models. And here, of course, we don't have any solution yet, but we are trying to come up with some solution how we can deal with this and address this. So we came up with this again, like 8-step. I don't know why we always have 8, but again, here we have 8 steps, which kind of promotes fair sharing of machine learning models, where we promote using standards and building Dockers and so on. So you can read the paper to get a bit more. Of course, this is initial study. We will still work on it. We need to still work on it, get more and more data, and improve things further here. Just a quick overview. Talking about interoperability, here we recommend using ONNX format, which allows you to kind of move things across. So you can have a model in TensorFlow, and you can move to PyTorch if you know the weights somehow encoded properly in the ONNX format, and do the inference. You can reproduce this, get the metrics correct, annotate the model, and disseminate them to BioModels so that others can easily use them. Here's another example. You can see there's a random forest model for predicting immune checkpoint blockade, and this model has been curated. As you see, we have all these metadata added on the right-hand side, manually curated, annotated. This makes the model discoverable, so people can search based on keywords and find the right models. They know these are the inputs of the model, these are the outputs of the model, and this allows you to kind of quickly search and find the models of interest. Again, Files tab has all the files that are needed, ONNX file being the main file here. And here in the last curation tab, you can see the figure which we reproduced. The figure on the top is the figure from the paper, and the figure on the bottom is the one which we reproduced. This allows, again, user to be confident and make sure that they reuse the model. Before they reuse the model, it is indeed the right model and it's doing the right job. With this, we have some sort of workflow to allow people to submit the model to BioModels and curate them and make them fair and reproducible and reusable and whatnot. However, we know there are other repositories like Hugging Face, and there are many other resources where you can have your model stored. So we wanted to make a slightly different workflow where you can store your model externally, but then share the metadata to BioModels. So the model is searchable through BioModels, but when you have to download the files, you will have to go to the other external resource and download it. And we worked out this with Elcilia, which is an external repository which has a good collection of machine learning models for antibiotic resistance. And these models are stored externally, but the models metadata are curated into BioModels, so they are discoverable. And once you discover the right model, you can click and go to Elcilia and download the models of your interest. This is again summarized on this in this article. If you like, you can go and read further or reach out to me. Right. With this, I would like to kind of bring a conclusion. So there's lots of effort to make sure we reproduce the model and make them publicly available, right? But this is not easy for us to do. It's really hard. So we want community to work together. So we had built this integration with Epicuron, a platform that allows crediting those who contribute. So it should be not just us curating. If someone else reproduces someone else's model, we're happy to take a look, cross-check, and then put that into BioModels and give the credit to those people. Right. With this, I would like to end. BioModels puts lots of effort in making models fair, making them findable, easily accessible, interoperable, reusable. And this means we are standing on the shoulders of a giant. We can pick up where someone has left and move forward instead of having to redo things again and again. With this is not the end. We wanted to focus more on more modeling approaches and try to provide standards and guidelines how to make the other modeling approaches reproducible beyond the 4 which I listed today. And another important news which I want to share quickly because I'm running out of time here is BioModel is moving. So thanks to Reinhard for giving us the opportunity to host in Florida, which is great. And we will have a new URL, which is biomodels.org. So people should start using this URL, and then you don't see much change afterwards. And I should especially mention TJ. He had been helping with all the technical migration, and of course, Tung from our team is of course also helping a lot in doing that. So what will happen in future? I mean, this is of course my models moving to Florida is a great news, but the way hopefully it will function in future is that it will be a co-hosting model where it will be co-hosted by Florida and another organization, hopefully where I will be moving. I don't have an answer yet where this will be. Then there are a few discussions going on. And also there will be a copy of all the models in BioModels collection within BioStudies in EBI. So EBI will be still in picture. We'll have archiving mechanism where we kind of back up all the models in BioStudies. And of course, Florida will be the main host right now, and probably there's another co-host sooner from another UK organization where I will be moving. Okay, with this, lots of people to acknowledge, and I finished the talk. Thank you for listening, and I don't know if that is enough time for questions. Otherwise, you can reach out to me.
Thank you so much, Sharif.